This protocol presents the process of preparing a large pool of human platelet lysate starting from platelet-rich plasma. By performing one freeze-thaw cycle, the platelets are lysed. Then, at least 10 units of human platelet lysate are pooled into one batch of pooled human platelet lysate, or pHPL. This volume of about 3 liters is further portioned into aliquots and again frozen. To deplete platelet fragments, these aliquots are thawed, transferred into vials, and centrifuged. The supernatant is the platelet fragment depleted pHPL, containing platelet-derived growth factors used to supplement cell cultures. The remaining platelet fragment pellet is discarded.
Hi, I'm Katharina Schallmoser from the laboratory of Dr. Dirk Strunk in the Stem Cell Research Unit at the Medical University of Graz. Today we will show you a procedure for the preparation of a pool of human platelet lysates. We will start from platelet-rich plasma, perform several freeze-thaw cycles, and then deplete the platelet fragments. We use this procedure in our laboratory to obtain a sufficient supplement for cell cultures as an alternative to fetal bovine serum. So let's get started.
This protocol begins with platelet-rich plasma, which is called PRP. This plasma might have been prepared by cytophoresis or derived from buffy coats. People outside the transfusion medicine field should purchase PRP from a local blood bank or another authorized provider. Check the sterility of the PRP unit by transferring 20 milliliters from each unit